AS Level Biology 9700, Paper 2, Variant 2, May June 2025. Question number 6, last video on this paper. Now the question number 6 is horseradish peroxidase is HRP is an enzyme that can be extracted from the roots of the horseradish plant Arborecia rusticana. HRP is used extensively in industry and technology in the reaction catalyzed by HRP. Hydrogen peroxidase is used to oxidize an enzyme substrate. This is summarized in figure 6.1 and the equation is organic substrate plus hydrogen peroxidase HRP oxidized substrate plus water. So question part A, inhibitors can have an effect on V maximum, the maximum rate of reaction, and Km Michaelis-Menten constant of HRP. Figure 6.2 shows the effect of substrate concentration on the reaction of HRP. So here in this case, the initial rate of reaction has been shown on y-axis and on x-axis substrate concentration has been shown. Now the question is complete figure 6.2. So by drawing a curve to show how the presence of a non-competitive inhibitor will affect the rate of reaction of HRP, use the curve you have drawn to obtain an estimate of Michaelis-Menten constant. So Michaelis-Menten constant, first of all, we have to draw. As you know, the substrate concentration initial rate of reaction, it will be slow downs. When the inhibitor has been non-competitive inhibitor is there, then what will happen? The rate of reaction, a little bit rate of reaction will be slow down. So because a non-competitive inhibitor works by binding to a site on the enzyme, that is not the active site. So it will bind itself on any place on the enzyme. So the binding causes a change in the shape of the active site, making the enzyme less effective at converting substrate into product, regardless of how much substrate is present. So the key effects of non-competitive inhibitor or reaction kinetics are V maximum reaction decreases since the inhibitor reduces the efficiency of some enzyme. The maximum possible reaction rate is lowered. On the graph, the new curve will reach a lower plateau. So Michaelis-Menten constant unchanged because the inhibitor does not affect the enzyme's affinity for the substrate. So can still bind to the active site. The Km value remains the same. So the curve will be changed. A little bit curve will be changed. So it will start right from here. So it will draw in this way. Little bit rate of reaction will be changed, but it will remain the Michaelis-Menten constant will remain the same. It will be it will be 0.06. So 0.06. So what I have told you, that is rate of reaction little bit will be changed. Active site will be changed, but the rate of reaction, smaller change in the rate of reaction. So, but Km value will remain the same. So it will be equal to 0.06. The next question. Scientists can design synthetic DNA nucleotide sequence to produce a synthetic HRP gene. These sequence will include a start codon and a stop codon so that translation of messenger RNA can occur. Explain what is meant by a start codon and stop codon. So start codon and stop codon will be three. First of all, start codon. Start codon, a three nucleotide sequence 3 nucleotide sequence on messenger RNA. Molecule that signals the beginning of the polypeptide chain. So it dictates the point where the ribosome complex should begin translation. And then the other stop codon is also R3 nucleotide sequence on messenger molecule that signals the end of the polypeptide chain synthesis. So it dictates the point where the ribosomes complex should stop translation and release the finished polypeptide chain. So next part of this question, part C, HRP is used in an immunological test known as sandwich ELISA. One use of this test is to diagnose disease. Figure 6.3 outlines the main steps in sandwich ELISA, in which a toxin released by a pathogen is detected in a sample of body fluid taken from a person who is ill. The test involves two types of monoclonal antibodies that can bind to toxin, capture antibody, and a detective antibody. So step one is prepare a test well by immobilizing the capture monoclonal antibodies, the sport surface. Second step, remove a sample of the body fluid from the person who is ill and add the sample to the test well. Step 3, wash out the test well to remove any substance not bound to the capture antibody. Step 4, add detection antibody to the test. Wash out the test well to remove an detection antibody that is not bound to the toxin. So add substrate for catalysis by HRP and add acid after a set time to stop the reaction. Now, after these details, the question is complete figure 6.4. Complete figure 6.4 to show the situation in the test well after step 5. So what will happen? The 8 detection antibodies, they will bind with the toxin. So they will be in this way. So they will try and attach themselves only. So this is HRP and they are detection antibodies. So they will attach themselves over there. So in this way, you will complete the diagram. So with reference to figure 6.3, describe one difference between the capture antibody and the detection antibody. The capture antibody and detection antibody. So this is the capture antibody and this is the detection antibodies. On detection antibodies, you can see the HRP is attached, whereas the capture antibody is without this. So now we have to write only one difference between them. So first of all, I would like to say that the key difference lies in their location and modification difference between capture and detection antibody. So capture antibody is immobilized to the spore surface to trap the target antigen toxin. It has no enzyme or label attached, whereas the detection antibody is added later and binds to the captured antigen toxin. It has an enzyme HRP attached to it. So allow for the which allows for the final color change. So the most concise difference we can say the detection antibodies has the enzyme attached to it while the capture antibody is immobilized on the spore surface and has no enzyme. So the detection antibody has the enzyme HRP attached to it while the capture antibody is immobilized on the port surface and has no enzyme attached. And the next question, figure 6.1, 6.5 outlines the reaction catalyzed by HRP in step 6. Step 6. So the organic substrate TMB is in excess and changes color when it is oxidized. This indicates a positive test result. TMB colorless, hydrogen peroxide, HRP. HRP is an enzyme which will oxidize oxidize the TMB into blue-green color, and the water will also be produced with it. So a student suggested that a low concentration of toxin may be diagnosed as a negative result instead of a positive result. So using a calorimeter after step 6 in the sandwich ELISA test would provide a quantitative result and help to avoid this error. With reference to figure 6.5, explain why using a calorimeter would provide a quantitative measurement for detection of a low concentration of the toxin and help to confirm a positive result. So we have to write this, its answer. So the first of all, I would want to explain that how chlorimeter is has an advantage. Use of chlorimeter has an advantage for low toxin concentration. So the key advantage of using a chlorimeter is its ability to provide an objective and quantitative measurement, especially when the color change is faint due to low toxin levels. Secondly, the chlorimeter measures the intensity of the color by measuring the amount of light absorbed by the sample. And according to the reaction in figure, the amount of blue-green product is directly proportional to the activity of the enzyme. So since the amount of HRP is proportional to the amount of detection antibody bound and the detection antibody is proportional to the concentration of the toxin captured, the absorbance reading is directly proportional to the original toxin concentration. So uh, when the toxin concentration is very low, the color change from TMB uh, oxidized is faint and difficult for the human eye to distinguish in the colorless state, so it leads to the potential error of classifying a low level positive as a negative result. So chlorimeter is far more sensitive than the human eye and can detect even a minute increase in absorbance above the background level. So by measuring the absorbance and comparing it to a calibrated standard curve, the student can objectively determine that a small amount of product has formed, confirming a true positive result despite the color being too faint to see clearly. This so validates a weak color change that a person might miss. So in this way, the chlorimeter has many advantages. So I would like to write. So the chlorimeter measures the intensity of the color, intensity of the color by measuring the amount of light, by measuring the amount of light absorbed by the sample. The key advantage of using chlorimeter is its ability to provide an objective and quantitative measurement. especially when the color changes faint due to low toxin levels. A colorimeter is far more sensitive than the human eye and can detect a minute increase in absorbance above the background level by measuring and absorbing by the by measuring the absorbance and comparing it to a calibrated standard graph. The student can objectively determine that a small amount of product has not confirmed a true positive result. So it was all about question number 6. Please like, share, and subscribe my channel. Thanks for watching.